Chapter 11, Mendel and the Gene Idea. In this video, part 1, we're going to be looking at these 3 you must know. So the terms associated with genetics problems, T1, F2, F1, dominant recessive, homozygous, heterozygous, phenotype and genotype, how to derive the proper gametes when working a genetics problem, and the differences between an allele and a gene. And I want to encourage you, actually, as you're reading this, to have your textbook nearby. There are some amazing diagrams in your textbook. I'm looking here, especially on page 221, figure 11.4. I highly recommend you look at that diagram on alleles, what an allele is, and that is a really, really great picture. And also figure 11.5 and figure 11.6. I'm going to be covering all these things in this video, but highly recommend. It'll help you so much to understand this stuff. This chapter is a lot of vocab, and a lot of it is probably review, hopefully some review. So I'd encourage you, again, pages 218 to 223 in your textbook. Just skim that, read it over, look at those vocab words and review it, and then use this video as like kind of a boost to your understanding. But textbook's really great for this chapter. Chapter 11, Mendelian Genetics. In this chapter, we're going to be looking at the research of Gregor Mendel, who was a monk who lived in the 1800s before we knew anything about DNA or chromosomes or meiosis. And his research was really helpful for us to understand some of the statistics and being able to guess if you have two parents, like for example, we have a purple flower and a white flower, we're able to predict what do we think the offspring might look like based on genetics and based on probability and statistics. So before Mendel, the thought was, okay, well, if you have a purple flower and a white flower and they reproduce sexually, the offspring will be like a blend of the two, kind of like taking two paints and blending their colors together and they mix. But what Gregor Mendel found was a little bit different. He found that if you took a purple flower and a white flower and you had them reproduce, well, they would produce only purple flowers, which is kind of interesting. And so his research led us to understanding of things like dominant and recessive, which you maybe remember from taking biology in the past. But in this video, we're going to dive into all of that, the probability, and what we know about how to make predictions of what an offspring will look like based on their parents' observable traits, right? And again, this is without any knowledge of DNA. So we will make some connections to DNA, but we'll take a look at basically like the research behind all of this. So let's start with who is Gregor Mendel? Well, he is the father of modern genetics, and it's sad because in his lifetime he didn't know. His work wasn't recognized until he died, but he worked in the mid-1800s in an abbey garden as a monk, and he documented inheritance. Inheritance means the passing of traits from parent to offspring, and he used pea plants. He used an experimental method, so he did the same thing many, many, many times, and he researched hundreds of different pea plants, and he collected his data and used quantitative analysis. He counted up. So he took pea plants, he had them reproduce, and he had offspring created, hundreds and hundreds of offspring. He counted them and he looked and he observed their traits. So purple flowers and white flowers, and how many offspring are purple, how many are white. And then he took statistics and he applied it to what he found. So he did research and then he found patterns in the data and he came up with an idea of, hey, maybe this is how we can predict what the offspring will be based on the parents' appearance. So we're going to take a look at how Gregor Mendel researched on his pea plant. But first, we need to know a little bit about how flowers reproduce. So flowers reproduce sexually, and in order to reproduce sexually, you need two gametes, egg and sperm. So let's zoom in here and let's take a look at how a flower were to reproduce. Well, there's actually two ways. So the first way is if a flower is true breeding. And you don't need to write this down. It's just going to help you later on when we learn more about the genetics of the pea plants, but just listen to see how the flower can make new offspring. So a true breeding plant would be if the flower actually reproduced with itself. And here's how it does it. So it actually has the stamen, which is kind of like the male part of the flower, and then the pistil, which is kind of like the female part, because the stamen actually has these orange pieces on the outside, which is pollen. And pollen are single cells and they're haploid cells. So these are the gametes of the flower. And if the pollen goes into the pistil here and reaches down to the egg, now you have egg and sperm, right? Or pollen, egg and pollen. And that's fertilization. Now you have your zygote. The zygote will divide through mitosis and make an embryo. And now you have a seed and it will develop into a whole flower, right? Kind of like a fetus developing in a human. That's true breeding flowers. And the offspring will look very similar to their parents because you have their own sperm and their own egg, right? But the other way is to do cross pollination, right? And the way to do that is actually if you took one flower and a different flower from a different plant. And you could take the pollen of this flower, and if it goes and reaches this egg, now you have cross pollination. Okay, so that's actually what Gregor Mendel did. So he took pea plants and he bred them and he made predictions based on what he found. So let's start here. So first, he cross-pollinated true breeding parents. So here are our parents. Here's the very, very, very beginning. And these are true breeding. So this white flower, for every generation before that, there's always been white flowers that bred with themselves. So he was certain that this was a true white flower. There was no way that on its own, if this reproduced with itself, it would produce a purple flower. The same thing here. This is a completely true breeding flower. Every generation before it has been true breeding and no cross-pollination has occurred. Okay, so he took these true breeding parents and he basically cut off the anthers of this purple one, so to ensure that it wouldn't breed with itself. And he took a paintbrush and he dusted off the pollen from this white flower and pasted it here so that it would fertilize this purple flower. He raised the seeds that developed and he observed the traits. And what did he find? He found that white and purple flowers, true breeding flowers, produced completely purple flowers. And you can count up all the purple flowers here, but he did this hundreds of times. Like he collected a lot of data and he found that every single time he did this, he got all his flowers were purple. And then this is called our first generation or F1. Okay, and these are hybrids. They're hybrids because they came from two different true breeding flowers originally, and they mixed. So cross pollination. Now, F1, the F stands for filial, and this is a Latin term for like son. So F1 is the first generation. Then he allowed these F1 generations to cross pollinate, so they exchanged their pollen with each other. And then he observed the next generation, the F2 generation. And what do you think he found? How many flowers were purple? How many flowers were white? Or was there a blend of both? Well, let's take a look at his results. So here's our P generation. So here's our original true breeding purple, true breeding white. After the first F1, all were purple. And then let's take a look at the F2 generation. Okay, so what he actually found was that in his research, he counted up 705 dominant, or in this case, those were purple, and 224 recessive, or in this case, white. So if you were to look out in the garden, mostly purple, but some white in that F2 generation. So all of these produce mostly purple and some white. And he boiled this down to a ratio of about 3-1. So for every 3 purple flowers, there was 1 white flower in the hundreds of offspring. Now, he did this multiple times. He took a pea plant with the pea generation, so true breeding yellow seeds, and then he crossed it with a true breeding green seed. And he found that the first generation exhibited the dominant form. So all the F1 generation would be yellow seeds, but then he crossed those F1 generations to get his F2, and he found, look, 6,000 yellow seeds, the dominant form, and 2,000 green seeds, which is, this again, a ratio of 3-1. You can keep going down the list and see the other traits that he was looking for in his research, and notice that every single time in the F2 generation, when you followed kind of the same thing, so true breeding in the parents, and then F1 are hybrids, and then the F2 is the cross-pollination of the F1 generation, he always found a ratio of 3-1. Okay, so he did this for, again, 7 different traits. And what did this mean? What did he find from this research? So let's look a little bit closer at what he did. So first, he took this parental generation. Remember, these are true breeding purple flowers. He crossed them with true breeding white flowers, and he figured out that the physical traits are called the phenotype. So the phenotype would be what you're actually observing when you're looking at the flowers. So true breeding purple, true breeding white. And he found that in the first generation, the F1 generation, he got 100% purple flower peas, right? We just talked about this. He allowed some of them to self-pollinate. And he found 75% of them were purple flowers and 25% white. And then again, that's the ratio 3-1. So that F2, filial 2 generation, is 3-1. Now, make sure you understand what P, F1, and F2 means, and that's one of your you must knows as well. So what did these findings mean? Well, what he found was that some traits actually mask the others. So the purple and the white flowers, they don't blend in the F2 generation. So you don't take a purple flower and a white flower and then get like a mixture, like a light purple flower. No, instead, the purple actually masks the white. So remember back here, so even though you had a purple and a white parent, for some reason, there's something about the DNA. Remember, they didn't know about DNA, but there's something about the purple flower that takes over in this first generation and masks the white flower. And then even here, there's something about the purple flower that's kind of taking over and masking the white flower. So that's kind of what Mendel found from this research. Now, he called the one that was fully expressed means show, what is seen, the dominant allele. And the recessive allele is the one that has no noticeable effect, or the gene makes a non-functional protein. And we're going to talk more about that as we kind of get closer into the research here. So let's take a look here at some chromosomes. Okay, so remember, when we're looking at plant reproduction, we're looking at a pollen, which is like a gamete. It's a sperm cell meeting up with an egg cell. And what do we know about what is inside the sperm and the egg? Well, we know that there are chromosomes inside which are haploid, right? They're haploid cells. And so when the sperm, when the pollen, and the egg come together, now you're going to create a diploid cell. If we zoom in really closely to the chromosomes, let's say this one was from the pollen and this one was from the egg, you'll see that on the piece of the chromosome here at the top, there is something called an allele. Now, what is an allele? It is a sequence of DNA. So if you were to take the sequence of nucleotides, so again, those are the bases that make up the DNA at this section, because remember, a chromosome is just a long piece of DNA that's tightly wound into a chromosome. If you were to look at the bases here at the specific locus, or this location right here, you would see a certain order. So maybe it's A, T, G, 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 right? And that is the allele that creates purple flowers, okay? Now, at this locus on the other homologous chromosome, right, maybe again, this is from the pollen and this is from the egg, you have a sequence of DNA here, and it's maybe A, T, T, G, A, like a different sequence if you were to unwind it here. And this would be the allele for white flowers. And we'll learn about this more later in later units, but chromosomes have DNA inside, and the DNA has the instructions to make proteins. And the proteins are basically what are going to give the flower its color or give it its pigment. Pigments are proteins. And so if the DNA has the instructions for building purple pigments, then the flower will look purple. If the chromosomes have the instructions for white pigments, then the flower will look white. However, in this case, we know that the purple is actually the dominant allele. So if you have the dominant allele, then the trait is going to be expressed in the phenotype. Wait a second, what did I just say? I said a lot of words. So remember, the phenotype is what you look like. So if I have a dominant allele, and down here we have the recessive allele, or the non-dominant one, as long as we have one dominant allele, the phenotype, the appearance of the flower, will look like the dominant allele. Okay, now, once again, we have two homologous chromosomes, one from mom, one from dad. These came together when pollen and egg came together for fertilization. And now you have the appearance. So this would be what the actual genes look like, but the actual appearance of a flower will be purple. So even though the flower has the instructions for white, it's not seen in the phenotype, because as long as you have the dominant allele, you will see the dominant trait. Okay? Again, here's another example. Here's homologous pairs. And remember, there are multiple genes on every chromosome. So this is a gene locus. So let's say, for example, we're looking at humans and we're looking at chromosome 1 of humans, right? Remember, we have 23 pairs, but let's look at the first pair. Here's the chromosome from mom and here's the chromosome from dad. So if I were to take this DNA and unwind it and look at the nucleotides, look at the bases of DNA, I might find the instructions for, let's say this is hair color. And let's say the sequence of DNA, A, T, T, G. gives me the brown hair, right? And so this gene is the gene for hair color. Allele, the capital G, is maybe brown hair. So this chromosome will make brown hair proteins. Whereas this chromosome from the father, it has a different sequence of DNA if you were to unwind at this locus, at this location, and this would be the recessive allele. So maybe this is blonde hair. Okay, so now as long as you have the capital G, as long as you have the dominant allele, your phenotype, what you look like, will be that dominant. So in this case, if we have allele and one blonde, you're going to get brown hair because you have the dominant allele. Now, at a different locus on the chromosome, maybe this is the eye color allele. This is still chromosome pair 1, homologous pairs. And again, you have a dominant and a recessive. Now, notice here that this chromosome doesn't have all dominant. So maybe down here, maybe this is like the height gene. So this piece of DNA has the instructions for how tall someone will be. And notice that the mother gave the short allele, whereas the father gave the tall allele. And so you will still be tall, but you got that from your father, right? So there can be a mix and match of like, it's not just like that the mom is completely dominant and her alleles and the father is completely recessive. It can depend. And remember, this is important when we talk about meiosis and crossing over, because right before the cell divides to make like sex cells, the sperm and the egg, these chromosomes will cross over and kind of swap their genetic material so that eventually the sperm cells, the egg cells, the gametes are going to kind of have a mix of these and everything kind of gets shuffled around through meiosis. So, what did Mendel's research tell him about how traits are inherited? Well, we learned that traits are inherited as discrete units. What this means is that for every trait you have, like hair color, eye color, skin color, they're inherited separately. So you will get your hair color and your skin color not from the same exact parent. They're different, right? Now, another piece that you need to understand is that for each characteristic or for each trait, a diploid organism, so we are diploid organisms, we have two alleles. We have two copies of each instructions for every trait, one from each parent. Why is that? Well, we have two copies of every chromosome. So remember, we have 23 sets of chromosomes and one from each parent, called homologous chromosomes. Analogy you can use is having like two editions of an encyclopedia. So you have like the Britannica and the Americana one, which are just like, they're basically encyclopedias that have the same information, but they may be slightly different in how they portray the information. In the same way, you have two homologous chromosomes, right? One from mom, one from dad. They have the same genes on them. They have the same instructions for certain traits like hair color, eye color, skin color. The difference is in the code. So this one might have the code for blue eye alleles, right? And this one might have the brown eye allele, and that's the difference between them. All right. Now, just because you have a dominant allele doesn't mean that it's better or more common. So, for example, you have two genes for this polydactyl gene. So, if you have two recessive alleles, so one from mom, one from dad, and they're both recessive, then you will have 5 fingers on each hand. But if you have at least one dominant allele, and one of your parents in their egg or their sperm passes down the chromosome that has a dominant allele, you will have an extra finger. So that's not very common, right? It's not very prevalent in the population. Actually, 99 individuals out of 1,000 only uh, have 5 digits, right? Like most of us. Most people are homozygous recessive, which we'll talk about later. This is two like lower cases alleles, so two little a's. And most of us are this. But if you have two dominant or at least one dominant and one recessive on your homologous chromosome pair, then you will have this trait. So the polydactyl allele is dominant to the recessive allele. So as long as you have it, if one parent passes that down to you, then you will get it. So some more vocabulary to look at. So some words that we need to know. So the difference between how an organism looks, so actually what you see, and its genetics, what its DNA looks like, have different terms. So the first term is phenotype. So when I use the word phenotype, I'm talking about a description of an organism's trait. So basically think about it like, this is not the best example, but a lot of times people describe this as like your physical characteristics, like what you can see. So like a phenotype would be flower color, right? That's what you can observe with your eyes. A genotype would be, okay, describing the organism's genetic makeup. Or in this case, we're talking about, okay, what are the alleles on each homologous chromosome for one gene? That's a genotype. So think genotype, think literally you're talking about what the DNA looks like, what the alleles are. Phenotype is describing what an organism looks like. So let's use Mendel's results and let's use the words genotype and phenotype. Okay, so what is the phenotype of this flower right here? Describe its traits. Well, a phenotype is it's purple, right? What is the genotype? What are the alleles that it has? That might be a little trickier. We'll come back to that. What is the phenotype of this one? White, right? What is the phenotype of this flower? Purple, right? Now, does this plant have dominant alleles or recessive alleles? Well, we know dominant because we know purple is dominant. What about this one? Well, it has recessive alleles because white is recessive. This one, does it have dominant alleles or recessive? Well, we'll come back to that because we don't really totally know yet what chromosomes this purple flower has. We'll talk about that in a second. Okay, so just another practice with this phenotype. So is what you observe. So here, right here, we have blue eyes and brown eyes. But the genotype, we're going to be using capital letters or lowercase letters to describe what are the alleles on the homologous chromosomes. So in this case, we have a lowercase b and a lowercase b. Now, when we use lowercase, we're talking about recessive alleles usually. And so what this is telling us is that on the two homologous chromosomes for the eye color gene, right, maybe this part of this chromosome and this part of this homologous chromosome, one from mom, one from dad, both of them have the recessive allele, the piece of DNA that says, hey, build blue eyes, blue eye color, build blue eye color proteins, pigments, right? But let's take a look at this eye. Our phenotype here is brown eyes. That's what we see. What's the genotype? What is the DNA? What does a homologous chromosome show? Well, it actually could be two different things. We'll come back to this a little bit. Perhaps one chromosome has the dominant allele and one chromosome has the recessive allele. Wait a second. If we have one recessive allele, why do we see brown eyes? Why do we not see a mix? Well, it's because the capital B or the brown eye allele is dominant. So even though you have one blue eye allele, you still have the dominant allele, and so the dominant allele will kind of mask or take over the recessive allele. Or maybe both of the homologous chromosomes both have the capital B allele or the piece of DNA that has the instructions for brown eyes on both homologous chromosomes. So when we're talking about genotype, we're talking about what is a sequence of DNA on two homologous chromosomes for a specific gene. That's really important. A genotype is what is a sequence of DNA on a homologous chromosome, on two homologous chromosomes for a certain gene. That's a genotype. We're talking about the alleles. We're talking about, okay, what actually is the DNA? Okay, so just like I talked about, we use letters to represent alleles, right? So for flower color, we're going to use a P, purple. So capital P or lowercase p. Okay, now the capital P is going to be our dominant allele. Our lowercase b is going to be a recessive allele. And just so I can make this like so clear, I want to show you what this means. So a capital P, again, remember, we're talking about chromosomes. If I'm looking at this locus of a chromosome, this might have the DNA sequence A, T, right? And this is simplified. It's probably a lot longer. But if you have this sequence, you are going to have, you're going to build purple protein, capital P. Okay, so purple. I don't have purple color here. If I were to look at a different chromosome, let's say this is the homologous pair. So this one came from the pollen. This one came from the egg. If I were to zoom into this chromosome and look at the DNA, I might see oops, I'm running out of space, T here. Oops, that's terrible. Okay, now notice a different sequence of DNA, right? AAT versus AATT. That's a different allele. It's a different instructions, different code. And so this might be the instructions for building white flowers. That's recessive, okay? Now, again, you have two of each because you're looking at the homologous pairs, one from mom, one from dad. So if you're looking at true breeding purple flower peas, remember, true breeding means one purple flower has been breeding with itself. It has taken its own pollen and fertilized its own egg for many generations. If it's a true breeding and it's purple, we have 2 capital P's because we'll learn about this more. If this was the chromosome that came from the sperm or the pollen, and this is a chromosome that came from the egg, then when you put them together, they're the same because we're working with the same plant, right? The same plant has the same alleles. True breeding white flowers would have 2 little p's, right? Because this chromosome came from pollen, this came from the egg, and they come together, but it's from the same plant, so it has the same instructions in the pollen and in the egg, okay? But what if you were to take these purple true breeding and purple white true breeding flowers and mix them? Well, you're going to get offspring that have a capital P and a lowercase p. Why is that? Why is our offspring going to be this genotype, right? Our genotype is capital and lowercase. Our phenotype, what it looks like is it actually looks purple. But why is this our genotype? We're going to talk about this more later, but the reason is this is on the homologous chromosomes in any of the purple flowers cells. So if you were to take one of these flower cells, you would find these homologous chromosomes, right? But when it's time to do meiosis, the plant's going to make gametes, right? And during meiosis, the chromosomes are going to be separated from their homologous pairs, right? Remember, meiosis, you have 2 homologous chromosomes, they make a copy, right? Then you have meiosis 1, separate. And then what happens to these sister chromatids, right? They separate. And so now in all the gametes, in all the sex cells, you have 1 copy of each chromosome, right? So if we're talking about the purple flower, so we have capital P and capital P, right, on the homologous chromosome pair. Make a copy, so all 4 of them have capital P, meiosis 1, meiosis 2. Okay, so now here's 1 gamete. Here's 1 cell. What does it have? What does a chromosome have? It's capital P still. Capital P, capital P, capital P. So if this is a pollen cell, it's a haploid pollen cell, and this is the gamete that gets fertilized, right? This is the one that goes in fertilization and mixes with the egg. Then this flower is going to donate a capital P allele. What about this one? When this white flower is going through meiosis, it only has 2 lowercase p's. So duplicate DNA, all 4 sister chromatids have lowercase p, meiosis 1, meiosis 2. All the egg cells here are going to be lowercase p. So if this pollen gamete fertilizes this egg gamete, then that's a zygote. The new offspring is going to have the pollens, capital P, and the eggs, lowercase p, and this would be their homologous chromosome pair, right? So if you were to look at those 2 homologous chromosomes, now you have capital and lowercase. Now think about, we'll talk about this more later, but what happens when this flower goes through, meiosis? Capital P, lowercase p, makes a copy, separates, right? Meiosis 1, meiosis 2. Now you have capital, capital, lowercase, lowercase. So their gametes are now mixed. So now depending on which of these gametes is chosen during sexual reproduction, could either be lowercase p or capital P, dominant or recessive. So let's say the lowercase p gets selected, then whatever the other flower combines with, that's now going to be their baby's offspring. Okay, so I know that's a little bit complicated. We'll talk more about it, but just kind of wanted to introduce you to that right now. Okay, so we've learned the words phenotype and genotype, and we know that 2 organisms can have the same phenotype. So this one, purple and purple, same phenotype, same appearance, same physical appearance, but can have different genotypes, different homologous chromosomes with alleles. So in this case, purple, we call this homozygous dominant. Why? Homo, the root word homo means the same. And zygous is kind of connected to the idea of zygote. Remember, a zygote would be sperm and egg, or pollen and egg coming together, right? So when the sperm and the egg chromosome come together, we make homozygous, same here chromosomes, okay? And we call it dominant because both of them are dominant. If you were to have a capital P and a lowercase p, the alleles now on the homologous chromosomes are different. And the root word for different is hetero. So here, this zygote right here is heterozygous because it has different alleles. But why is it still purple? Why is it not a light purple? Remember, because the capital P, the dominant allele, is dominant. It's taking over, it's masking the recessive allele. So let's apply what we just learned to Mendel's experiments. Let's remember Mendel took two true breeding flowers, so a true breeding purple and a true breeding white. This is a parental generation. Cross them. Now we have the F1 generation. Here's our hybrids from our true breedings. And the phenotype, what do we see? We have 100% purple flowers, right? That's the phenotype. Then we have them self-pollinate, and we have our filial 2 generation, or F2, when the hybrids self-pollinate. And now you see 75% purple phenotype and 25% white phenotype. And remember, if you look at this as a ratio, that would be a 3-to-1 ratio. For every 3 purple flowers, there's 1 white flower. And remember, Mendel did this like hundreds of times. So based on all his 100 pea plants that he researched, he counted in the field, he found, or even thousands of times, he found this was like the pattern that was coming out. But let's apply some of the terms. So this is a phenotype, but what's a genotype? Well, because this is a true breeding flower, it's never really crossed with a white flower. So it's only been reproducing with itself. And so it's always going to produce purple offspring because its egg and its pollen, its egg and sperm are always producing P alleles, right? And they're always combining capital P's, right? But the white flower is true breeding recessive, or we call this homozygous recessive, homozygous dominant, right? What is our first generation? Well, this flower can only donate a capital P. This flower can only donate a lowercase p allele in its homologous chromosomes for the zygote here. And so each of these flowers, or we actually would look at like hundreds of flowers, but 100% of them would actually be heterozygous. So one capital and lowercase, because this one donated maybe pollen, that was capital P allele. This one donated egg, lowercase p. Okay. But then what is our genotype for our F2 generation once we take the hybrids and we have them self-pollinate? So if this flower were to donate its pollen to this flower's egg, what would be the offspring? So just take a moment and think, how would we be able to figure that out? How would we be able to figure out what is the chromosome, or what is the allele on the chromosome that's being donated in this flower's pollen? And what is the maybe egg cell, or what is a chromosome in the egg that could be found in this flower's gametes? And then what would its offspring look like? Ooh, here comes the fun part. So how can we figure this out? We can use something called a Punnett square to predict the probability if we took a heterozygous hybrid here and a heterozygous hybrid here, and we were to have them sexually reproduce with each other. So this one's maybe this one's sperm, or this one's pollen, and this one's egg. So how do we do it? Well, we're going to imagine that after this purple plant does meiosis, it can make only 2 different gametes with different alleles. So let me draw this out for you because it is pretty complicated. And oops. Okay, actually, let's do this. If you're going to take my pen here. Oh, much better, much better. Okay. So let's say we have this purple flower, okay? And if we were to look inside one of its cells, we would find homologous chromosomes, right? And this one would be capital P, and this would be lowercase p. Oop, that's kind of hard to see, but capital lowercase. What does that mean? That means this piece of DNA at this locus has the instructions for building purple pigment. This piece of DNA at this locus has the instructions for building white pigment, but because the capital P, the purple, is dominant, we're only going to see purple in the phenotype and the physical characteristics. Now let's talk about meiosis, right? Remember, in meiosis, we make a copy. So now you have 2 P's, capital P's here on each sister chromatid, and 2 lowercase p's here, right? Meiosis 1, they're going to separate, and they're going to go like this. So now you have capital P, and you know what? I'm just going to go erase this. Sorry, guys. I'm just going to go capital and capital. But you know, when I'm drawing these capitals, I'm talking about the alleles on the chromosome. Okay, and then this one is lowercase, lowercase. So what have we done? This is 1 cell, and meiosis is 1 cell. This is meiosis 1. Okay, then remember, meiosis 2, we're going to split this down in half, right? going to split this in half. And what's going to separate are the sister chromatids, right? I didn't draw the chromatids, but now you know. You can have P, P, capital P, capital P. This one splits in half and has a lowercase p and a lowercase p. Okay, and remember, these are individual cells. So this, this is terrible. This is a gamete, this is a gamete, this is a gamete, this is a gamete. So let's say these are sperm cells or pollen cells, right? What gametes can this flower pass? Capital, capital, or lowercase. So really we can kind of distill it to really the plant can only donate a capital or a lowercase. So that's why we put a capital here and a lowercase here, because we're talking about the potential alleles that can be passed in each gamete. So this is one, let's say we call this a sperm cell, or this sperm cell. Depending on which one's available during reproduction would determine which one is selected. So this plant actually has all sorts. Oh, I have some people. Hello. Wait, okay. I'm gonna go say hi. Some of you guys are at my door. Hold on. Okay, where were we? Punnett squares. Okay, so let's take a look at the other side of the Punnett square. And what this is showing us is this flower right here. Okay, so this is going to be, if we're talking about sexual reproduction, this is going to be the plant that donates a sperm. This is going to be, sperm is going to come land on the flower and it's going to fertilize this flower's egg. Okay, so this is this side. And then the same exact thing as this, right? You have your capital P, lowercase p. This is the genotype of the homologous chromosomes found in every single one of this cells, right? These are this is diploid. It's two copies of the same chromosome, right? Same genes, but different alleles. Heterozygous, different alleles here. And when it's going through meiosis, first it's going to make a copy of each chromosome. So capital Q, capital P. Then meiosis 1, we have one cell over here. Oops. We have the homologous chromosomes are going to separate. Lowercase p, and that's meiosis 1. And then meiosis 2, now you have capital, whoops, that's capital, lowercase, right? Oops, and let me see. Yeah. So this is one cell, and one cell. Now, plants make like so many gametes, right? Like this plant has like thousands of sperm cells. So the plant goes through meiosis many times. So even though in one round of meiosis you make 4 pollen cells, the plant has way more pollen, right? But they will do it many times. And so whichever pollen cell ends up landing on this one will be the pollen that's fertilizing this one. So it could be the lowercase p or the capital P. It's kind of random, whichever one is the one that ends up landing on the other flower. And then for this one, it's whatever egg is available at that time, it will be the one that's fertilized. So it could be this capital P or the lowercase p. So that's why we put the eggs here. These are the optional alleles found on the homologous chromosomes in this flower here. Okay, that was a lot of detail. Hopefully you understand Punnett squares a little bit better. So how do we do the Punnett square? You might remember this from taking biology. But basically, you're going to take these alleles and you're going to bring them down. So you're going to bring it all the way down. So P down, P, capital P. And then same thing here, bring the little p down. And for this, maybe I'll use a different color. So this is a little p. Okay, so we've done that. And then you're going to go across here. So capital P. And then just a convention, you always put the capital P before the lowercase. Okay, so then across here, a lowercase p, lowercase p. So what is this showing us? This is showing us if this is a sperm, if this is a pollen that is fertilizing this flower, and this is the egg that's available at that time, this will be the genotype of the offspring of the child flower. Same thing here. If this is the allele found in the gametes, sperm and egg, then this is going to be the offspring, or this, or this. Okay, so that is what a Punnett square is showing us, showing the potential offspring. Now, it's not that these flowers are only going to have 4 offspring. It's if these flowers had hundreds of offspring, we're going to find that this is going to show us the probability that you're going to get homozygous dominant babies, heterozygous babies, or homozygous recessive babies. Got that? Okay, next. Oop, so then it's just filling in. Let me erase this. Clear screen. Okay. So again, this is what we find. So our genotype is going to be homozygous dominant, but our phenotype is purple. Our genotype is heterozygous, but our phenotype is purple. Our genotype is heterozygous, but our phenotype is purple. Our genotype is homozygous recessive, but our phenotype is white. So yes, you can have two purple flowers, and some of them can be white. Why? Because each flower donated an allele, a gamete, that was recessive. So if you have two heterozygous parents, and they ended up donating in their sperm or their egg their recessive allele, you could have this offspring with homologous chromosomes that have homozygous recessive genotype. Okay, so we can line up our results. So these are our offspring, and we can actually do some calculations. So what is the percentage of, oops, someone's coming in. Okay, back to what I was saying. So percentage of phenotype. What is the percentage of purple flowers here out of 4, just to make it simple? We have a 75% purple flower to 25% white flower phenotypic, we call it phenotypic ratio, or 3-to-1. But what is the percentage of genotypes here? Now we can use our Punnett square to predict. So out of these 4, what percentage are homozygous dominant? If you said 1 out of 4, or 25%, you'd be correct. 2 out of 4, or 50%, are heterozygous, and 25% are homozygous recessive. So our ratio here is 1. So for every 1 purple, there's going to be, for every 1 homozygous dominant, there's going to be 2 heterozygous to every 1 homozygous recessive. So if you simplify this 1 to 2-to-1. Okay, so once again, homozygous means same alleles. Homo means same. And it could either be 2 dominant of the same alleles or 2 recessive. Heterozygous means different alleles, capital and lowercase. So putting everything together, our genotype, if you have a homozygous dominant genotype, 2 of the same alleles, your phenotype is going to be purple because that dominant allele is taking over. Heterozygote is going to be 1 dominant, 1 recessive, but the phenotype, the appearance, is still purple. And then the homozygous recessive would be 2 lowercase alleles. And again, we're talking about the genotypic ratio. We're talking about 1:1 because there's 3 different genotypes, right? There's homozygous dominant, heterozygous, or homozygous recessive. So that's why you have 3 numbers in this ratio. When we're talking about the phenotypic ratio, we're looking at just 2 things. We're looking at purple flowers or white flowers. So every 3 purple, we have 1 white. Okay, now is it possible to determine the genotype of an organism by looking at it? So if you just saw this purple flower, could you be 100% certain that it's homozygous dominant or heterozygous? How would you figure it out? So what you could do is actually a test cross. So you could crossbreed the dominant phenotype. So here's our dominant phenotype and an unknown genotype with a homozygous recessive. So we're going to take this here and we're going to cross it with something that we know. We're 100% certain that this white flower is homozygous recessive. Why? Well, we know that it has to have 2 little p's in order for it to be white. If it had a capital P, it would automatically be purple. And if it had 2 capital P's, of course, it would be purple. So we already know the identity of this white flower. And so we're going to take this, we know it's purple, right? We have a dominant phenotype, but we don't know its genotype. We're crossing it with a recessive. So let's do a test cross. So let's take this one. We don't know what its genotype is, but we're certain that this is little p, little p. So let's say if it was, we're going to make a prediction, a hypothesis. If it was homozygous dominant, then we could do our Punnett square. So let's take our, this maybe is our sperm. So we take this sperm is going to, or this pollen is going to land on this, egg here. So we're literally going to dust, we're going to take a little paintbrush, dust off the pollen and place it here. And the only alleles in the gametes, these pollens, can be little p. Same thing here. The eggs can only be capital and capital. So when we do our Punnett square and we fill in, we get 100% heterozygous, right? Or all purple flowers. Okay, purple phenotypes. But if you were to take a purple flower and cross it, right? We don't know what its genotype, we're just predicting here. Cross it with a white flower. We're going to put our pollen here, and we're going to put our egg. So this one can either donate a capital P or a lowercase p. Then our offspring, let's say we had these reproduce like hundreds of times and we counted up all the offspring, you would find 50% are going to be purple and 50% white, right? So 1 to 1. So if you did this, remember, imagine you did not know the identity, the genotype of these flowers, but you did cross them, and you actually find a bunch of white flowers, then you're like, oh, wait, yep, this one's heterozygous because it's hiding this little p allele in here. And we find that in the offspring. But if you find that all the flowers are purple, then you're like, oh, yep, this one had to have had 2 capital P's because it was never able to donate a lowercase p to its offspring to make a white phenotype.